Okay, and good morning, everybody. So, as some of you might notice, I'm not Jason Stein. Unfortunately, he starts feeling sick yesterday afternoon, and this also holds true for the complete rest of the speakers. So, unfortunately, we only have videos. So, yeah, we have to relax and simply watch the videos. There's a small intro from Jason at the beginning, and then we'll keep going on with the videos. Therefore, we wrote down their contact information. Everybody of those people agreed, feel free to write them emails, and so they're really sorry, but unfortunately it's caused by COVID and other reasons. So yeah, let's start with the intro.
Hello everyone, welcome to our symposium called Imaging and Analysis of 3D Cellular Resolution Brain Structure Using Tissue Clearing. I'm Jason Stein, I'm the organizer from UNC Chapel Hill. So the motivation for this symposium is that recent advances in tissue clearing and light sheet microscopy are enabling cellular resolution imaging of brain structure and model systems such as mouse brains and human brain organoids, and are now being extended to post-mortem human brains. So this symposium will discuss methods for tissue clearing and antibody labeling of human brains, novel microscopy techniques to rapidly image tissue cleared brains at cellular resolution, methods to analyze these large-scale images, and application of tissue clearing to measure the differences between diffusion-weighted and tissue-cleared axonal fiber orientations. So we have three excellent speakers, as well as myself, who will be telling you about their research in tissue clearing. So the first lecture will be from Jennifer McNabb from Stanford on comparing MRI microstructure maps and tissue clearing 3D histology in the same human brain specimens. Our next speaker will be Zhuhao Wu from Mount Sinai, who will talk about HubClear. It's a simple, adaptable, and scalable clearing method for human brain molecular and cellular profiling over large volumes. Next will be Elizabeth Hillman from Columbia, who will talk about HOLIES, or human brain optimized light sheet microscopy for cell type resolved mapping of entire human brains. And finally, I will give the last presentation on cellular quantification in tissue-cleared whole brain mouse images and human cortical organoids. So I hope you enjoy the symposium, either on this virtual platform or in person in Glasgow. So thanks.
Hello, everyone. My name is Jason Stein. I'm an associate professor at UNC Chapel Hill in the Department of Genetics and the UNC Neuroscience Center, and I'm happy to share with you today some of the work we've been doing in my lab on cellular quantification in tissue-cleared whole brain mouse images and human cortical organoids. So causal genetic factors are starting to be identified for different neurological and psychiatric disorders. Identified genetic variants that create risk for a disorder are just starting points in understanding what causes these disorders. Base pair changes in the DNA do not directly alter human behavior. Instead, they must be mediated through some molecular, cellular, and systems level cascade. My lab attempts to make a contribution by understanding how disease-associated genetic variants affect gene regulation, cytoarchitecture, and macroscale brain structure. Much of my previous work has been on mapping genetic effects on macroscale brain structure as measured through MRI. In this type of study, we need a population of genotyped individuals where we have measured their genetic variation, as well as images of macroscale brain structure where we measure variability in brain size. Within the ENIGMA consortium, our recent project sought to identify common genetic variants influencing cortical surface area and thickness. We acquired MRIs and genome-wide genotyping from 60 cohorts across the world, comprising almost 34,000 participants, with replication in an additional 17,000 participants. We identified hundreds of loci in the genome where variation is associated with interindividual differences in cortical structure. But these intriguing findings always leave us with another question, and that is, what is the cellular basis underlying these differences in gross anatomy? Is it changes in the total number of cells in individuals of different genotypes, changes in the number of cells of a given cell type, changes in the overall size of cells or their branching patterns? And unfortunately, MRI does not have sufficient contrast or resolution to answer these questions. So we've recently turned to different techniques. So today I'm going to talk to you about how our lab is studying how genetic variation influences cortical cytoarchitecture. That is how cells are organized in the brain. And we do this, we measure the brain cytoarchitecture through tissue clearing and light sheet microscopy. So there's been a recent surge in new methods for imaging intact brains at cellular resolution without the need for physical slicing. These are called tissue clearing methods and allow the full mouse brain to be imaged intact. We've imaged a mouse model of neurofibromatosis type 1, a disorder which often presents with intellectual disability and autism, to understand how this mutation alters brain structure to lead to risk for a neuropsychiatric disorder. We've used a mouse model that knocks out the NF1 gene specifically in progenitor cells of the developing cortex using an EMX1 Cree flox design. These mice then have both copies of their NF1 gene disrupted, only one copy of their NF1 gene disrupted, or no copies of their NF1 gene disrupted. We acquired 6 mice in each of the different groups. We then use iDisco tissue clearing technique to be able to image these and immunolabel them with markers of lower layer neurons, which is marked by CTIP2, upper layer neurons marked by CUX1, or all nuclei marked by TOPRO3. These were then rapidly imaged on a commercially available light sheet microscope. So this is an example of what a tissue cleared mouse brain hemisphere looks like. At the top of the image is the rostral side of the brain where the nose is, the lateral side where the ears are, and the back here of the brain, you can see the bright folds of the cerebellum. So this image is quite zoomed out, but labeled with TOPRO3, which is a nuclear marker. So here you can see that the brighter the image is, the higher the density of the nuclei. So you can see like in regions here of the hippocampus, there's a really dense number of nuclei. So zoomed in on the right side here, you can see that these images provide sufficient resolution for visualizing individual nuclei in the brain of a P14 mouse, both in XY and XZ. So as all the neuroimagers at this conference know, analysis of images requires a detailed processing pipeline. So we went over the top 3 here. A graduate student in my lab, Oleg Krupa, who recently graduated, developed a new pipeline for generating and analyzing these images on a commercially available light sheet microscope. This pipeline includes image stitching and alignment across different channels, registration to a defined atlas that is well annotated, as well as quantification of features within the image, which in our case is nuclei, and then to quantify these nuclei and co-localize them with different markers for different cell types. After that, we have maps that show the differences in cell counts between different brain regions here. So here's what these images look like. There we go. With the right side being the NF1 conditional knockout and the left side being controlled. So here you can see labeling of 2 different layers of the cortex, the lower layer and the upper layer. We also have a nuclear stain, which we use to count every cell. We're able to count cells within each region of the cortex, which is identified through registration to an annotated atlas. And you can see those registered regions here. And so as it finishes going through the cortex, you can see that it zooms out here in this video, and you can see the NF1 conditional knockout has somewhat of a bigger brain compared to the control. So these images are quite beautiful, but of course we need to count features in the image in order to make quantitative comparisons. So in order to quantify nuclei, we first needed to train a computer what a nucleus looks like. Ole, the grad student leading this project, with the help of some undergraduates in the lab, manually labeled over 50,000 nuclei by hand to train a deep learning model called a UNet. Ole then applied that UNet to the images and was able to quantify all the cortical cells, as shown here. So each dot represents the centroid of a nucleus. The dots can be seen here. And we compare these automated labels to independent manual labels and found them to be very accurate compared to other methods. So they were compared to two methods, ClearMap and Cubic. What the program calls as a nucleus is usually a nucleus, so it has relatively high precision. Although the program does miss some nuclei in densely packed regions, which is evidenced here by somewhat lower recall. So also the more training that we provide the program, shown by the number of images as input, the better the algorithm does to call a nucleus. So overall, nuclear classification can be done in P14 brains using a deep learning technique. So you can see that this technique can be used to count all the cells in the cortex, as well as to co-localize with markers for different cell types, upper or lower layer neurons. And each dot in this video here represents a different cell type, or sorry, a different cell. And then zooming out, you can see the entirety of the cortex and then labeled quantified nuclei for the upper layer neurons or the lower layer neurons here. So what did we find? So here you can see a representation of a flattened cortex, the nose at the top here, the cerebellum at the back here. So using Numorph, we measured the average cortical thickness of cortical regions and detected increased thickness in the posterior regions, as well as a slightly, we detected increased thickness in the posterior regions and slight cortical thinning in the orbitofrontal regions here. So these differences bear a strikingly similar pattern to human MRI findings where patients with neurofibromatosis type 1 were shown to have thicker occipital and thinner frontal cortices. Next, we found that the overall increased thickness was associated with increased cell count, as shown by these TOPRO3 here, but not from excitatory neurons, as shown by this CUX1 or CTIP2 positive, instead by non-excitatory neurons, which must be glia. So in sections, we found that there was an increase in astrocytes, marked by GFAP, and oligodendrocytes, marked by OLEG2, that explain this increased thickness. So here we think that we have explained in part the cellular basis underlying MRI findings in human, though it would be good to, of course, to confirm this in post-mortem human tissue from neurofibromatosis type 1 patients as well. So overall, thickness increases were driven by non-neuronal glial populations. Though the thickness findings are similar to what was found in human, we also were able to explore differences in cell type proportions that would not be visible on MRI. We found a very interesting set of areal differences in the NF1 cortex. There was a depletion in the proportion of upper layer neurons in more medial regions of the NF1 conditional knockout as compared to wild type. There was also a decrease in lower layer neurons in more lateral regions of the NF1 conditional knockout as compared to the wild type. So there seems to be a reciprocal change across the cortex where depletion in the proportion of upper layer neurons is associated with an increase in lower layer neurons. We thought that these findings were quite interesting given that slice immunohistochemistry at different regions may give you quite different results. So this emphasizes what we think the utility of the 3D approach of imaging. So since the publication of our article, we've been expanding into several new research directions. So one of them is expanding our training set through manual annotation. Oops. One very important thing to consider is that our algorithm to count nuclei is only as good as the data set it was trained on. So increasing the size and the scope of the training set will be essential to improving counting accuracy, especially in difficult regions like those that are in densely packed areas like the cerebellum, the hippocampus, or embryonic brains. So we have in the lab 15 undergraduates who are now doing this annotation using a tool that's been developed by a visualization engineer, David Borland, who's our collaborator. The goal here is to trace tens of thousands of nuclei in order to have an even better algorithm. We are importantly tracing the entirety of the nucleus here rather than just doing one dot per nucleus. And this will enable us to do instance segmentation, so we have the full shape of each nucleus rather than just counting a nuclei. The tool we developed is called Segmentor, and we call our nuclei labelers nuclei ninjas, with the original thought being that this would be like Fruit Ninja, where you sort of have to separate two nuclei that are falsely attached to each other. So we currently have 14,000 entire nuclei annotated through the dedicated work of our nuclei ninjas and getting more and more every day. Current manual annotation takes a lot of work though for a single user, as each block that we give our undergraduates contains roughly on the order of 1,000 nuclei. So it usually takes an undergrad an entire semester to segment one volume, considering that they usually contribute about 10 hours per week. So our goal here was to, we have another goal, which is to make it easier to make progress on a given volume, given that we want more users to contribute. So we're hopeful that we can get thousands of manual annotators, but each doing far less work. So maybe just a few minutes of work instead of a whole semester's worth of work. To do this, we need to move away from our standalone application, Segmentor, that I previously showed, and from segmenting the entire volumes. Instead, we need to assign tasks in parallel so many people can be editing a given volume simultaneously, but not fighting each other for annotations. So we're implementing this through a web-based framework that we're calling Ninjato. That's now being tested, and we hope we'll be ready for prime time by the end of this year. So the instance segmentation is now being implemented and tested, and we have reasonable precision and recall now, especially for the P14 mice, where we have a larger training data set and where nuclei are spaced farther apart. So we're actively increasing our manual labeling in the P4 mouse to increase performance there. The real advantage, again, of this instance segmentation will be that we can calculate not only the counts of the nuclei, but also features of nuclear shape, like the volume of the nuclei that may be useful for identifying cell types without extra labels. So this work is being done by our collaborators, Gurang Wu and his postdoc, Junbo Ma. Okay, so switching gears a bit, I'm going to talk to you about applying some of these tissue clearing techniques to a different model system of the brain. In this case, human brain organoids, which are 3D cultures of brain cells that mimic some of the structure of the developing cortex. So human cortical organoids can be derived from individuals who are deeply phenotyped. And in this case, we are making cortical organoids from individuals of the Infant Brain Imaging Study, or IBIS, who have had MRI scans and behavioral measurements longitudinally since infancy. It's a very cool and detailed cohort. So from those individuals, we are acquiring blood samples, isolating peripheral blood mononuclear cells, overexpressing reprogramming factors to turn the cells back in time to an embryonic-like state, which is called an induced pluripotent stem cell. And from there, from the induced pluripotent stem cell or iPSC, differentiating them into cortical organoids, getting whole organoid imaging and being able to relate that back to the MRIs from that same individual to see how well they model the individual's cortical growth. In this way, we're trying to assess the fidelity of organoid cultures. So we generate iPSCs from this population and differentiate them into cortical organoids, which are these little balls of cells. The entire protocol takes around 3 months and involves culture in a mini spinning bioreactor, which spins around. This mini spinning bioreactor is shown here and spins around to increase media diffusion and to prevent cells from dying in the center of the organoid. So after 3 months, there's the spinning. So after 3 months, we fix the cells and again perform the tissue clearing and imaging. We're also getting other measures of these organoids as well, but for today I'm going to focus on the tissue clearing. So here's an example of an organoid differentiated in our lab. The image of this organoid was obtained using iDisco tissue clearing and light sheet imaging to image the full structure of the human cortical organoid at cellular resolution. We label in different colors markers for progenitor cells, the developing lumen, dividing cells, and all nuclei measured in TOPRO. So here's a video of the organoid, so spinning around and then going through slices of the organoid. Each of these sections represent neuroepithelial buds, which are like the closure of the neural tube. So each one of those is like sort of a new developing brain. So you can see here a lot of neuroepithelial buds, and these are the expected cell types and morphology of cells, showing that we are indeed generating models of the brain in a dish. So we have applied NuMorph to count cells in organoids, and this seems to work reasonably well for neuronal markers like CTIP2, shown here, and then new morph counts here, and CUX1 shown here, and new morph counts here. So these label lower layer neurons and upper layer neurons. However, cells that are really densely packed, like the neuroprogenitor marker PAC6, do not seem to be accurately segmentable with our current combination of imaging resolution and our segmentation tools. So for progenitor areas, what we end up doing is measuring the size of the lumen, so marked by NCAD surrounding the PAC6 positive cells. So this can be thought of as analogous to the size of the developing germinal zone, which contains the progenitor cells that eventually differentiate to neurons to form the cortex. So we sort of have a two-layered approach here. For neurons, we calculate things differently than we are where we measure neurons, we measure individual cell types, whereas progenitors, we're measuring volumes. We do have some preliminary data here showing that we can correlate the size of the progenitor-like zone in vitro with the overall cortical surface area in vivo. So in a small sample size of only 9 donors right now, who had both MRI at 24 months of age, where the organoids were generated from iPSCs of that individual, we find a relationship between the size of the germinal-like zone in the organoid with the actual size of the in vivo cortex. So you can see that relationship here. This is somewhat expected given that a greater number of progenitor cells in the developing cortex is expected to lead to a larger size of the adult cortex. So far this was only conducted in 9 donors, so we are increasing our sample size for this study. If it holds up, it would show though that organoids are a high fidelity model system and can be predictive about the size of brain structure in the future from that individual. We aim to use this study to understand the cellular basis for brain overgrowth in autism. Okay, so overall, we've developed tools to quantify cellular brain structural differences in 3D images of mouse brains. Using these tools, we found that an NF1 conditional knockout mouse had similar spatial patterns of cortical thickness differences to the human, and that increased thickness is driven by glial cells. We are expanding these studies by increasing our training data set, and we're generating iPSCs and cortical organoids to model cortical development in individuals who have been densely phenotyped from birth. Overall, tissue clearing technologies provide complementary measures to MRI to understand how genetic variation leads to differences in brain structure. So we are applying these tissue clearing technologies in multiple model systems to understand how genetic variation leads to risk for complicated neuropsychiatric disorders. So I really want to thank the people who have contributed and done all of this work. So especially I would like to thank Ole Krupa, who was recently PhD'd, developed this new morph package and worked with our collaborators, Mark Zilka and Lei Jing. I'd also like to thank Rose Glass, who's shown here, who generated all the organoids, and our collaborators from the IBIS consortium, who have allowed us to generate these cortical organoids from their study. So thank you very much for your time and attention.
Okay, as I said, so there will no questions since nobody's here. Yeah, let's simply continue with the next video.
Good morning. My name is Jennifer McNabb. I'm an associate professor at Stanford University, and I'd like to thank the organizers for the invitation to be a part of this session. I'll be speaking today about comparing MRI microstructure maps and tissue clearing 3D histology in the same human brain specimens. The development of MRI methods for mapping properties of tissue microstructure continues to be an extremely active area of research. Instead of mapping non-specific diffusion tensor parameters, there is a strong focus on developing ways to access highly specific and biologically relevant features, such as axon diameters, myelination, neurite density and dispersion, and even a recent paper aimed at capturing some sensitivity to changes in glial morphology. Improvements in MRI hardware, such as stronger, faster gradients, are really helping this field evolve. More robust and efficient encodings enable a more diverse set of encodings to be acquired with a higher level of precision. And this provides a richer data set that can hopefully support more sophisticated models of tissue microstructure. Still, we are always relying on indirect measurements in MRI, and therefore multimodal comparisons are essential to evaluate whether we have successfully achieved the specific sensitivity to tissue features that we are aiming for. One approach to evaluating MRI microstructure maps is to perform MRI scans on post-mortem tissue and then subsequently histology in the exact same tissue. In this way, it's possible to capture information about some of the histological components within an MRI voxel. Conventional histology usually involves sectioning the tissue and mounting very thin slices on slides. The tissue components of interest are identified using either a chromogenic or fluorescent label, and then the tissue slides are imaged with light microscopy. Unfortunately, some microstructural brain features are difficult to capture with 2D histology. For example, axonal fiber bundles and blood vessels project in 3D, and the curved nature of the cortex can make it difficult to obtain slices of tissue that accurately reflect the distinctive layering patterns. This recent review paper highlighted some of the difficulties associated with inferring on 3D microstructure from 2D samplings, showing convoluted structures, complex distributions of tissue components, and the possibility of failing to capture tissue components that are sparsely distributed. Still, it is possible to generate 3D volumes from 2D histology. This paper detailed some of the strategies to make this work and also highlighted some of the remaining challenges. Adjacent tissue slices can be distorted differently when placed on the slide, and here they demonstrate an approach to better align the slices. Still, they show some residual level of misregistration. For this reason, there's interest in performing histology on intact volumes of tissue. Tissue clearing aims to reduce the opacity of tissues such that optical imaging of intact tissue is possible. When combined with fluorescent labeling, it allows for whole brain 3D imaging with subcellular level specificity. Now, tissue clearing is a century-old approach that has been reinvented with recent developments in reagents and protocols, efficient fluorescent labeling, and rapid volumetric imaging by light sheet microscopy. Tissues are opaque because different components of cells have varying refractive indices, which cause a strong scattering of light. And so the tissue clearing process involves removing lipids, which are a primary source of this scattering, and also various solutions that enable refractive index matching. There are many, many different protocols for tissue clearing, but generally they all have these steps: fixation of the tissue, some sort of chemical treatment, removal of the lipids, labeling, refractive index matching, and microscopy. I'm going to show some results using the Clarity protocol first, and this involves embedding a hydrogel in the tissue, which helps to lock the proteins and nucleic acids in place. And the lipids are then washed out using a detergent. There's a stain of the proteins. Proteins or nucleic acids are labeled with fluorescent markers, and imaging is done using light sheet microscopy. Now, these images are very exciting. We can see a whole mouse brain here, which is about the volumetric limit that we've been operating at. Here we can see a whole mouse brain with exquisite visualization of individual neurons. We also seen some very nice results in larger tissue samples of a rhesus macaque, showing some of the expected patterns in the visual cortex, and again here with the very easy to recognize Gennari stripe. The original Clarity paper did demonstrate the technique in human tissue, although there are some notable differences when working with human tissue. Human tissue tends to be even more heavily myelinated, and therefore if passive clearing with detergents is used, it can take a very long time. Further, it's not possible to rely on transgenic labels, and many of the antibodies needed to visualize features of interest are quite large, and it's non-trivial to have them propagate evenly throughout the tissue cuboid. Still, Nicholas Weisskopf's group demonstrated the ability to use a number of different types of stains in cleared human brain tissue, and also the ability to stain fairly evenly over several millimeters of depth. When comparing MRI with tissue clearing data, the MRI must be performed first because the elimination of the lipids and perhaps other aspects of the tissue processing pipeline do eliminate most types of MRI contrast. When cutting the tissue, we found it essential to perform MRI acquisitions and photography at each stage of the cutting, such that the frame of reference relative to the whole brain is not lost. Now, imaging fixed post-mortem tissue does involve a number of important considerations. It's been shown that the post-mortem interval, the time between death and fixation, is best to be kept short. This is showing that the axial diffusivity does get longer as that post-mortem interval is longer, gets longer. Fixation is achieved with immersion fixation for human samples as opposed to perfusion fixation with animal samples, and that means it'll take at least 6 to 8 weeks to usually fix all the way through a whole brain, or that can happen over a much shorter time frame if you're just using a much small section of tissue. T1s and T2s are stable in the fixed once the tissue is fixed, but they tend to be much shorter than compared to in vivo, and proton density is lower. It's also been shown that the T1 and T2 weighted contrast between white matter and gray matter changes, in fact, inverts in some cases. And so that's a consideration for any structural imaging. And also because you're imaging at room temperature, you have a much slower diffusion rate, means you require more diffusion weighting to get the same level of contrast. In terms of an experimental setup, usually the brain is surrounded with some sort of non-protonated fluid, a perfluorocarbon. The idea is to have a susceptibility match surrounding the tissue, but also not to have too much signal from the fluid surrounding the tissue. There's a desire to eliminate air bubbles. Sometimes we put it in a vacuum overnight to do that. Also because of the susceptibility difference with air pockets. And you want to pack it in a way so that it's physically stable in the scanner, but also not squished so much that it deforms the shape of the general shape of the brain. It's important to keep it temperature stable because this can impact our diffusion and our T1 and T2 measurements. Running the scanner for long periods of time can sometimes heat up the bore. You also ideally would like a nice tightly coupled RF receive coil, and the MGH group showed a nice example of this recently. There's also the consideration of the availability and cost of potentially several days of scanning if you want to do high resolution diffusion imaging. The shorter T2s and the stronger diffusion weighted needed means it really does take quite a long period of time to get these types of data. And then there's also issues just in terms of keeping the scanner running continuously. These scanners aren't set up to do so, and there can be lots of pop-up warnings, and you can run the risk of gradient overheating from running it continuously for so long. There's pesky SAR monitors, which aren't really relevant to the postmortem tissue, but can be difficult to turn off. And remote monitoring can be really, really helpful to run these types of experiments. In terms of acquisition strategies, there's no motion, and there's some changes in T1 and T2 to consider. The fact that T1 and T2 changes have meant that balanced SFP, which has this funny T1 over T2 contrast, provides really good gray-white matter contrast. And so balanced SFP with some phase cycling has shown to work really well to get really high-res, beautiful structural images in postmortem brains. For diffusion imaging, there's no motion, so you can use these segmented acquisitions to get really high-resolution, low-distortion images. And you do need B values about 3 times higher than you would use in vivo to get a similar level of contrast. Still, if you take these things into consideration, you can get absolutely drop-dead gorgeous, beautiful data that has a lot of value on its own, even before you compare it to any histology. Now, in terms of the types of stains we looked at with the clarity data, we focused on a small molecule stain, the pyridium iodide, which stains the DNA and RNA and is fairly easy to use. We also used a neurofilament stain, which is requires a much larger antibody, but provides a nice visualization of some of the fiber structure. And we wanted to look at that because we felt like we have a fairly good idea of how to map fiber orientations with diffusion imaging. And that was a good place to start with these comparisons. A really big part of these comparisons is getting an accurate alignment between the MRI and the optical images. We found that using the vessels as landmarks, both for guiding the registration and evaluating the accuracy of it, was incredibly useful and gave us a lot of assurance that there was a close correspondence between the two types of images, and we could do a very detailed voxel-to-voxel comparison. There's also a need to extract in some sort of quantitative way the parameters, histological features from the histology data. Luckily, there's pretty well-defined ways to do this for fibers, fiber structures. There's the structure tensor analysis, which we were able to do in 3D with this data, and relies on image intensity gradients to identify the boundaries of fibers. We also spent some time looking at the volume over which you look at these image intensity gradients and how that relates to the resolution of your microscopy data and how that affects the ultimate fiber orientation distribution that you estimate. And it turns out it is quite important to think about that closely and think about the size of the structure within the histology that you're trying to capture and how that relates to image resolution and how that might relate to your MRI data and the sensitivity you have to different sizes of features in the MRI data. This is an example side by side of the clarity neurofilament stain with the structure tensor estimates at each location of each MRI voxel, and then a diffusion tensor estimate of the principal diffusion orientation at each of the MRI voxels. And the color map is showing the angular difference between those two orientation estimates, showing in the blue, very good agreement, low angular difference. Some disagreement around the edges of the sample, but generally a pretty close correspondence. We also looked at a comparison of diffusion tensor orientation estimates and orientation estimates in the clarity using structure tensor, but on the vascular, the clarity data that shows the vasculature rather than the neurofilaments. And this was interesting because there's been some open questions about whether the vasculature contributes at all to some of the white matter orientation estimates that we get with diffusion. And in this case, we didn't see any correspondence between the two. It is interesting though, because it has been shown that the vasculature does tend to run along similar orientations to a lot of the white matter tracks. And there's some open questions about how this might impact some fMRI measures as well. We've also experimented a little bit with some iDisco in human tissue samples, which is a different type of tissue clearing protocol. The lipids are generally dissolved rather than washed out with a detergent. And this seems to work pretty well. And we've had some success looking at melanin concentrating hormone and also myelin basic protein and DAPI and neurofilament stains. And it seems like another promising approach for doing these types of comparisons, particularly in human tissue. This is lastly, I just want to highlight this really nice study led by my collaborator, Casey Halpern. He was very interested in looking at the connectivity of the nucleus accumbens. Dysregulation of the nucleus accumbens is implicated in a number of neuropsychiatric disorders, and there's an interest to treat this area directly with deep brain stimulation, but the initial results have been kind of variable. And one hypothesis is that the nucleus accumbens is quite functionally heterogeneous, owing to different parts of the nucleus accumbens connecting with different parts of the brain. And so Casey was very interested in us to look at this with tractography and see if we could segment different parts of the nucleus accumbens based on different tractography evidence of different connections with different parts of the brain. And then here is also an example of using clarity to try to highlight some of these different connections and the orientations of some of the pathways entering and exiting the nucleus accumbens. And also a very nice comparison with some of the tractography results in the same sample. And then this is in vivo data where we did do a tractography-based segmentation based on differing connectivity profiles of different parts of the nucleus accumbens, which seemed to line up quite well with some of the known histology. And what was really beautiful was another additional affirmation of this segmentation was some results from a deep brain stimulation that was performed. And you can see retrospectively here, we aligned the tractography-based segmentation to show whether the electrodes were in the two different tractography-defined parts of the nucleus accumbens or in the same tractography-defined section. And you can see on the left side, on the top image, there's two electrodes that are in different parts of the tractography-defined sections of the nucleus accumbens. And in that case, they saw a very differential response when they stimulated from each of those two electrodes. Whereas on the bottom, when they appear to both be in the more inferior portion, they showed a much more similar response. So it's a really nice sort of multimodal study to try to answer a very specific hypothesis. So in summary, MRI capabilities for mapping biologically relevant parameters continue to evolve. Direct comparisons with advanced histology, such as tissue clearing 3D histology, provide new opportunities to improve the interpretability of MRI biomarkers. I want to thank my group members and my funding sources and the organizers and participants here again for the opportunity to participate.
Again, sorry, we can't take questions because she's not here. So let's simply continue with the next two talks and try to see them.
Hi, I'm Zhuhao Wu from Mount Sinai in New York. First, I want to thank the organizers for this great meeting and this inspiring symposium. Following the last exciting talk, I will discuss more details for tissue clearing techniques on human brain samples. Together with our collaborators that you will hear more today, we aim to leverage on tissue clearing and light sheet imaging to map out every single cells in the entire human brain. For this audience, I don't need to emphasize how important yet challenging it is to profile the entire human brain with cellular details, covering its many anatomical compartments and the enormous cell type diversity and connectivities within. We have seen many exciting progress to better comprehend the entire mouse brain, but the human brain is 1,000 times, to be more accurate, over 2,000 times larger than the mouse brain, with many other challenges that I'll touch upon later. Even the relatively small mouse brain is not that easy to study in its entirety. We have been developing and applying tissue clearing techniques to make it fully transparent, as you can see here, so we can acquire fine details inside using fast light sheet imaging without the need of physical sectioning and laboratory construction. If we can scale this fast approach to the entire human brain, then we can hope to finish within a reasonable amount of time. Another challenge is that unlike mouse brain, with many available genetic tools and reporters, we need to rely on labeling endogenous markers for the human brain samples. Here, I want to cover the general procedures of our tissue clearing. It sounds like a magic to make a biological tissue fully transparent, but it is indeed following basic physical and chemical principles that I'll discuss. When light travels through a biological tissue, it will get deflected or affected by the cell membranes. It's not just a plasma membrane, but also the extensive membranous structures inside the cell, like the Golgi network and various organelles. Therefore, most light will get scattered off after just a few cell layers. In order to make a tissue fully transparent, we first need to remove the membrane lipids. After complete debullition, a fixed tissue essentially becomes a nearly homogeneous protein scaffold. That if we can match its reflecting nets with a high reflectiveness buffer, then it becomes essentially fully transparent, or now the light can travel through it without any problem. Over the years, we have been optimizing our procedure following this general principle that I just mentioned. For delimitation, we have been engineering our lipid extraction buffer that can solubilize both the hydrophilic head as well as the hydrophobic tail of the polarized lipid that are enriched in biological tissue, while preserving the protein scaffold in its native form. For refractingness matching, it is indeed difficult to measure a brain's refractingness directly. However, we can extrapolate 100% hydrated BSA to be about 1.5 to 1, based on its linear relationship of the concentration and its refractingness. Using this number as a starting point, then we can approach that a brain's refractingness is about 1.5 to 9. I do want to point to you that the brain's refractingness may depend on the actual procedure. During our optimization of the procedures, we have been trying to delimitate the sample completely while preserving the protein scaffold in its original form, or therefore the overall refractingness would be fixed to a very narrow range that we can match it really well. With this said, we have been optimizing and developing our tissue clearing protocols through iterations over the years with my extended collaborators. Showing here are some examples done by our original protocol IDISCO that are still being widely used in the field for robust clearing and labeling various tissues covering brain and other organs to profile the fine anatomical details inside. I do want to point out that IDISCO do shrink the tissue a little bit, and for the brain, it shrinks the cortical region a little bit more than the subcortical region. This issue is addressed in the second version of our protocol, IDISCO Plus. A brain image with IDISCO Plus is well preserved in its morphology so that the HOMA imaging can be registered back accurately to its original MRI scan or to the standard ALICE. This enables us to develop automatic pipeline for brain alignment registration and quantitative analysis, which is critical for handling large datasets generated by the HOMA imaging. We have further enhanced the lipid extraction procedure in our protocol so that it can not only clear brain, but also fat tissue, which is the most lipid-enriched organ in our body. Hence, this protocol was called AdipoClear. AdipoClear can directly extract triacylglyceride, the major lipid depot in the fat. Brain also has a trace amount of triacylglyceride as a normal metabolic byproduct. It could be rapidly accumulated during injury and aging. Therefore, if you use AdipoClear to clear a brain, you can also get more superior clearing performance. Together with other improvements, AdipoClear preserves the tissue antigenicity very well, so that now majority of the IHC-validated antibody is compatible with our HOMA procedure. Because our clearing is following on general principles that are not only well conserved across different organs, but also across different species, therefore, we can combine all our recent advancement in tissue clearing and directly apply for the human brains. Also, our procedure is based on chemical engineering. So, in another word, all that we need to do is find a bigger jar with a human brain slab into it and run through the same chemical procedures, and then you'll become transparent like what you can see here. So this is a medium-sized human brain slab from 89 years old donor after 4 months formalin fixation, and it still worked quite well. We are currently optimizing and adapting the human brain clearing procedure for a whole human brain imaging pipeline with cellular resolution. Of course, this is being done in collaboration with a team of great scientists. Our Human Brain Bank Director, John Query, will help to screen and procure human brain samples in the best conditions we could. Then Alan Zephyr will take the best prepared sample for MRI imaging. MRI imaging will be utilized for 3 major purposes. With help from Bradley Delman for radiology reading, we'll understand the pathological status of the sample. Then the MRI imaging will serve to incorporate the MRI-based ALICE to the sample that we're going to process. Finally, MRI imaging template will serve to incorporate the later high-resolution human brain slab imaging to the original brain space. Putting all these together, we can map fine anatomical and pathological details in the brain sample that we're going to process with help from Patrick Hoff. I'm not going to talk about the imaging, which will be done by our great collaborator Elizabeth Human at Columbia, leveraging on her super fast light sheet imaging system. and with her extended collaborators at Columbia for initial data processing. The data will be deposited at BIO, which is led by Alex Roglowski's team. What I will be focused on today is talking about the sample preparation. We'll take the 5 ml thick brain samples, turn it into fully transparent and labeled format to feed into the imaging pipeline. We have optimized our HOMA labeling procedure for human brain samples. As you can see on the top, here is a Norse labeling, and you can appreciate the sparse cell body distribution over centimeters in the human brain block. And bottom left here is MPY labeling for another distinct neuronal subclass. Not only you can appreciate the cell body distribution, you can also see their extensive neural projection in both gray matter and white matters. In the bottom right here, green labeling every single nuclei, while the magenta is the arteries coursing through. As you can see that the labeling is even and complete over centimeters in the entire human brain block. We have identified dozens of markers that could label diverse structures in human brain samples, while we are planning to screen many more monoclonal antibodies to cover over 500 neural targets for compatibility. Such kind of data set is not currently available in the field, which we believe is a major limitation for people to adapt for human brain hormone IHC. We'll share our result online. We focus on monoclonal antibody, of course, it's because it's reproducibility, but also because it's scalability, given their renewable reagent, which is critical for providing large samples like human brain, among other factors that I'm not going to discuss today. Here is our antibody screening pipeline. We'll take the publicly available monoclonal antibody resource that everyone can have access later and screen it over mouse and human brain samples, utilizing our optimized 6-section IHC protocol. We use our 6-section IHC protocol because it's a nice bridge between the standard histology to HOMA application. So our screening result will not only have a good predictive value for the later HOMA success rate, but also it can serve as a good reference for broad IHC applications. We choose the top candidate from the validated monoclonal antibodies and do a HOMA processing on mouse brains as a proof principle. Both our extensive 2D categorization as well as 3D profiling on selective targets will be released through our website portal. From this data, we'll try to design the best strategy to label human brains in HOMA. This would project to generate a much larger database, as Elizabeth will discuss later today. Here are some of the data you'll be able to find from our web portal currently, covering early screening result of monoclonal antibody on mouse brain samples. As we are ramping up our project currently, we hope to upload more data covering both mouse and humans soon. Eventually, from our web portal, you'll be able to find both curated monoclonal antibody for diverse cell types and neuronal structures with 2D or 3D reference dataset. More importantly, you'll be able to find our detailed tissue IHC protocol. We believe detailed protocol is critical for users to successfully implement the resource that we're going to generate. Here are some of the recent results that we screen cell type markers on human brain samples. Along this process, we are also optimizing the human brain IHC performance to see what will be the best optimal result for each of the given marker. As you can see here, I'm using relatively short-fixed human brain samples. What I have seen over a battery of different markers that human brain samples fixed for 3 days or 4 degrees generally give me the best performance. After knowing what we should expect, we have also incorporated a HOMA antigen retrieval procedure to work with long-fixed human brain bank tissues. In general, longer fixation will start to decrease the sensitivity of most of the monoclonal antibodies. Some are more sensitive than the others. With HOMA antigen retrieval, we are able to restore many of the markers to its expected performance compared to the short-fixed standard. We hope this effort will help us to achieve quantitative analysis of human brain sample with IHC, with understanding of various conditions that might influence the IHC performance outcome. Eventually, we hope to utilize the battery of markers we're establishing here to not only understand how the cell type composition is across different brain regions, but also understand how they may dynamically change during development agent in disease. We hope that these new tools and resources that we're building for cellular and molecular profiling of entire human brains will enable new possibilities for us to understand how the brain structures is being composed of, how brain functions, and how these functions may be disrupted in various diseases. I'll stop here, and I want to acknowledge the members of my group for the large amount of work that they have been doing to push forward the development of this exciting technical platform. We hope that the tools and resources that we're currently building will help facilitate the field for molecular and cellular mapping across the entire human brain. I also want to acknowledge my great collaborators, especially the Holy School. It has been so exciting to work with all of you. And my funding support, this project is largely supported by the Brain Initiative funding. Finally, I want to thank for all of you listening to my talk. I hope next time we'll get to meet in person and have more extensive discussions along this project. Hope you enjoy the rest of the exciting talks and have a nice day. Bye.
Good morning, everyone. My name is Elizabeth Hillman from Columbia University. I'd like to start by thanking the organizers for the opportunity to present this work, and particularly to Jason for putting together this symposium. I'm going to be talking about human brain optimized light sheet or HOLIS microscopy for cell type resolved mapping of entire human brains. And these are my financial disclosures. So the National Institute of Health Brain Initiative Cell Census Network has done incredible work over the last few years, mapping out, using all sorts of new technologies to map out connections and cell type distributions throughout the mouse brain. And although this has taught us a huge amount about the brain and how it works, it's a fair question to ask, what can we do next? How can we take what we've learned here in the mouse and all of these new technologies and translate it to getting new knowledge about the human brain? So as we were really intrigued by this opportunity to think of what we could do in the human brain, we realized that what we wanted to do was try our best to push to not just image pieces of the human brain at cellular level, but to be able to image the entire human brain. Now, why would we want to do that? Well, each human brain is completely unique. It's shaped by a lifetime of experiences, genetics, and diseases. And so to us, the most compelling challenge is to understand differences between different brains and get a handle on what is the variability from person to person. So everybody's brain is different. It's got a different shape. It's going to have different aspects to it. And it's understanding those differences that we felt would bring the biggest insights. So for example, do cell type distributions differ from person to person, or do they change as a function of development? Do differences in one place get compensated for by differences in other? So although we can learn a lot from in-depth studies of different regions and subregions, in order to grasp variability and enable comparisons and to create a database that would allow us to then take any piece of brain from any person from any region and be able to go and compare it to a sort of standard brains, we want to be able to capture the entire brain of each individual human. And so again, the point here is to embrace the fact that there is this variability between the brains and not to seek to just generate some atlas that represents one average human brain. So how are we going to do it? Well, we have this incredible technology called tissue clearing in microscopy, which allows us to actually take a piece of tissue, make it transparent. And you've already heard about this, I'm sure, in the earlier talks. But the big difference here is we have this intact three-dimensional chunk of tissue, and that means we don't have to worry about doing very thin sections and repeatedly imaging lots of different pieces and kind of trying to stitch them all together. We can capture this kind of beautiful three-dimensional structure in intact tissues. So this technology has been really refined and works great for mouse brains, and their kind of smallest dimension is about 5 millimeters. But mouse brains are really small. And also what you're looking at here is a video of an expressed genetically encoded fluorescent protein, which was actually inside of the mouse. And so therefore we didn't actually have to stain this sample. So we need to be able to convert this technology to something that's going to actually work in the human brain, including to clear the brain as well as to stain it. Now, you probably just heard from my colleague, Zhu-Hao Wu. He is brilliant at this, and he has developed this hub-clear method, which allows us to take chunks of human brain, basically slices about 5 millimeter thick, and he can convert them into being beautifully transparent, as you can see here on the right. He's also spent a great deal of time looking at different types of antibody staining, so basically doing immunohistochemistry in human brain and screening many, many antibodies to get different types of staining. So down here on the left is NPY, up in the middle here is nitric oxide synthase. So if we can actually process these tissues, and the idea is to keep them within sort of 5 millimeter thick slabs of the entire brain, and then image each of those slabs sequentially, and to stain all of those for different cell types, we would be able to image the entire brain at cellular resolution. But how are we going to do the actual imaging? So when we started to calculate this, we realized that the human brain is 2,000 times bigger than the mouse brain. And it's also already pretty difficult to image a whole mouse brain. So the standard ways that people use microscopes, this is depicting confocal scanning microscopy. So if you've got a resonance scanning confocal in your lab and it can go at 30 frames per second with 1 micron pixels, it would take well over a year of continuous acquisition to collect that data. And each pixel would only be seeing the light for 30 nanoseconds, which would mean your signal to noise would be really, really bad. And so you would use up all your time in your microscope core. But light sheet microscopy is a little bit different. So instead of just illuminating one point at a time, it illuminates an entire sheet all at once. This makes it much, much more light efficient, less phototoxic, and it enables much, much longer integration times than point scanning. However, the geometry of light sheet microscopy is typically quite awkward, and most light sheet microscopes aren't optimized for high-speed imaging. Now, fortunately, my lab for the last almost decade now has been developing technologies for really high-speed 3D light sheet microscopy. And here's just some crazy images that we've collected over the years, looking at all different things from fruit fly brains to crawling fruit fly larvae, C. elegans, worms crawling around, beating hearts, and down here in the bottom, looking at things like living mouse brain, looking at neural activity and blood flow changes. And so we decided to test this technology out on cleared tissues, and here's some beautiful images that Creeper in my lab was able to acquire incredibly quickly, and that made us realize that this was a technology that we may be able to translate into being able to image the human brain. So, sorry, there was a video there. This is an example of the data when you zoom in on just this little section up here. So as I mentioned before, this is the standard geometry for light sheet microscopy, and that's not too bad if you have just this sort of small mouse brain about the shape of a small olive. Now you can take this geometry and rotate it in order to be able to image, for example, a more sort of contiguous thick slab. However, these geometries and these working distance of these lenses limit you to being able to image only about 2 millimeters into tissue. And the more sections that we have to have, the more edges we have to deal with, and the more complexity we have to deal with. So using what we call our scape geometry, but here we've renamed it Holus because we're going to just keep this sheet statically still, we can actually image the intact thick section over 8 millimeters in depth if we need to. And we basically illuminate with an oblique plane of light, and then we detect that light back through the same objective lens. So it looks a little bit like this if we just put a cuvette of some fluorescein here underneath the objective lens. And so the way that we're going to be able to collect this data is we'd have the 5 millimeter thick cleared brain slab here actually mounted in a cassette with index matching glass either side and then oil on the top. And then we simply move the brain slice around at constant velocity, and we'll be collecting these kind of cross-sectional slabs of data that we can then reassemble into the entire slab, which we can then reassemble into the entire brain. So here's an example of that data collected using a prototype system. And you're going to see now this is the angle at which we collect it. So each image is an oblique angle through the sample, and then we literally just move the sample along. What you're looking at here is white nuclear labeling. The red is labeling the vasculature, and the little kind of cyan dots are the nitric oxide synthase expression. And so we can collect all of these colors at the same time in our system, and that's a big aspect of us being able to go fast, is to be able to do lots of spectral multiplexing. And I just want to point out here, so the other really important thing, and you'll see this a little bit later, is this ability to label all the nuclei. So you can see that every single nucleus of every single cell has been labeled and is easily distinguishable from each other. And then if you look at how the nitric oxide synthase expression overlays with that, you can see the way in which we would be able to detect whether or not a cell was expressing a particular protein. Now, we can do some calculations about how much data we're going to generate here. This is a kind of a complicated table, and the point is that, you know, we're just embarking on this project now, and we want to make sure that we don't, you know, either overdo it or underdo it. So in a lot of our pilot tests, we're looking at what kinds of resolutions, what kinds of color ranges we need, and so on. But this one's a reasonably good example here. We would be able to get 8 different colors simultaneously acquired with reasonably good spatial resolution, certainly sufficient to see those nuclei and resolve different cells. We think that we could get the imaging time down to about 8 days, which is a huge difference from what we would get if we were doing confocal microscopy. And that's where it's really, really important. We wanted to be able to commoditize this imaging to the point where you could reasonably expect to do multiple whole human brains, not have this be one project where you get one single brain and then you can't sort of do anything with it. The other important thing to look at in this table is that we estimate that this data set would be about almost 3.5 petabytes in size. And that's really important for us to consider here as well. So the analysis pipeline then is a really key aspect of this, because we don't want to generate 3.5 petabytes of data that's just going to sit on a server and nobody's going to be able to do anything with it. So we need a streamlined and efficient pipeline that is going to allow us to process and share this data so that people can actually learn from it. So the data I just showed you shows the kind of general format of what we're expecting to have. So the little white dots represent the nuclei, and then these different colors represent the different channels that we would be detecting with different antibodies. So what you can see here is that actually this color encoded mixture here that I have at the top is actually just a linear mixture of this, as I've just colored it, red, yellow, and blue set of different antibodies. And so it's relatively simple mathematically to find the location of all of these little white dots. That's actually something that can be done with relatively simple image analysis algorithms now. We're working with a team at Columbia in machine vision who are implementing this incredibly quickly. And then so what you can do is by finding the locations of these cells, then you can look through all of the different color channels and then basically index which what is the expression pattern of proteins, so what is the proteomic characteristic of that particular cell type. And something really interesting happens here if you have overlap between your different protein expressions, because actually what you can get is a code for each cell. And so in some cases, we hope that this will significantly increase our dynamic range to detect different types of cells. If you look down here, for example, if we even are really conservative here and we just say we have 2 levels of either there's no expression or there's some expression, then we have 7 colors, we could be able to resolve between 128 different combinations of antibody. And in this case, if we can just detect whether there's none, some, and then a lot, that could allow us to encode over 2,000 different combinations and thus 2,000 different cell types. Now, this would be done in a very data-driven way, so you would have this kind of vectorized information where you know where each cell was and what its different proteomic expression levels were. But then you would be able to go back in that data to the original data and then start looking at things like cell type morphology. We think that this is a good strategy because it would allow us to create this kind of vectorized, table-based data, which will be much, much smaller and much more accessible to people for quantitative analysis. Then, of course, the last part that's really important is that we put this onto some kind of common coordinate framework, because we want to be able to take the cellular level data and project it back onto things like standard atlases, and also project standard atlases down onto our data set. And so in order to do this, we're going to be taking intermediate scale images of these cassettes that are going to be easily registered. We're going to look at also doing some slide-free sort of block face imaging of the slabs. And we'll be able to use these diffeomorphic transformations between these different spaces. And so we want to, again, keep everything in this coordinate system space so that we can always go back and forth without having to duplicate this data or create any kind of one single giant TIFF file that includes all of the data. And so we're working here with the team at Mount Sinai, Dr. Alan Seifert, looking at doing high field MRI on the ex vivo brains right after they have been fixated, and then working with Mike Miller's team at Johns Hopkins for doing these transformations between the different atlas spaces. And so this is the final kind of rundown of what this pipeline looks like. So we can actually get whole brains, again, from Mount Sinai's Neuropathology Brain Bank. We're going to do high field MRI. Then we're going to slice them into 5 millimeter thick sections, going to perform that delipidation, staining, index matching process, then image them, and then we're going to pipe all of the data into the brain imaging library at Pittsburgh Supercomputing Center to run those algorithms. So I just finally want to thank the team. This is a big team effort, and we're really just getting started on it. Juha and I are the lead PIs on this. We have this team at Mount Sinai, the Brain Imaging Lab, Columbia University, and these guys down here helping particularly on the image analysis and data sharing side. And then this is the team in my lab right now who are working on all of the things that you just saw. With that, I want to just thank everybody else in my lab, as well as my institute and the rest of my funding. Thank you very much for your time.
Okay, yeah, I'm sorry again that we can't take questions. I think there should be the contact information. Please feel free to write emails to the speaker. They definitely would have liked to be here, but unfortunately it didn't work. So have a good day.